Welcome to the deep dive. This is where we unpack those really dense academic papers, pull out the key stuff and just give you the core knowledge.
That's the plan.
So today we're diving into a really big question in modern conservation. Is the shiny new tool always better than the tried and tested methods?
Yeah, it's a great question. And we're digging into a specific paper to get some answers. It's a 2021 meta-analysis from Ecology and Evolution. The lead author is Fediajevaite.
And the title kind of gives the game away, doesn't it?
It does, but it also hints at the complexity. Its meta-analysis shows that environmental DNA outperforms traditional surveys, but warrants better reporting standards.
Okay, so it outperforms but. That but sounds important.
Correct, very important.
So that title really sets up our mission today. We're looking at environmental DNA, or eDNA, trying to figure out if it really is the consistently better tool for biodiversity monitoring that, a lot of people seem to think it is. I mean, the promise is incredible, isn't it? Like, reading an ecosystem's history from just a cup of water sounds like science fiction.
It really does. And for anyone maybe less familiar, eDNA is basically just genetic material left behind by organisms. Think shed skin, feces, mucus, stuff like that.
In the environment.
Exactly. So you collect samples,
Yeah.
water, soil, maybe snow, extract the DNA, and then you can figure out which species are present. It works for everything from tiny zooplankton right up to big mammals.
And it's popular because it doesn't harm anything.
That's a big plus, yeah. It's non-destructive, and it can be incredibly sensitive.
Sounds revolutionary. So why isn't it used everywhere all the time?
Well, that's the thing. It is revolutionary, but its practical, approved application is still quite narrow. Everyone sees the potential for conservation, but it's only formally signed off for monitoring a few specific species globally.
Like which ones?
Good examples are the great crested newt in the UK, or invasive species like bighead and silver carp over in the US. Those are cases where the method's been really thoroughly validated and, crucially, standardized. But they're kind of the exceptions right now.
Okay, let's dig into that. What's the promise that got everyone so excited in the first place? Why do researchers seem to love eDNA?
Well, the big selling points you always hear are, one, it's often more sensitive than traditional surveys. You can pick up things you might otherwise miss.
More sensitive.
And two, it's frequently cheaper. And that's obviously a massive deal for research projects or conservation groups that are often running on tight budgets.
Cheaper and more sensitive. Sounds great. Any other advantages?
Yeah, definitely. Well, like we said, it's non-destructive. You're not disturbing the animals or their habitat. Plus, you sidestep what's called morphological identification bias.
Ah, so you don't need an expert biologist to visually identify, say, a tiny little fish larva.
Exactly. Or some obscure insect. If you can read its DNA barcode, you know what it is. Takes away that potential human error in identification.
Okay, but like any new tech, there must be downsides, right? What are the pitfalls? Where can it go wrong?
Oh, absolutely. There are definitely limitations. You've basically got two main types of risk. First is false negatives.
Meaning the animal's there, but the test says it isn't.
Correct. And that can happen for lots of reasons. Maybe the animal's life cycle means it's not shedding much DNA right then, or the population density is super low. And critically, the environment itself plays a role.
How so?
Well, you can get things called PCR inhibitors in the sample.
Oh, yeah.
Think of stuff like humic acids from soil. They basically gum up the works of the molecular machinery we use to copy the DNA.
So if you can't copy the DNA, you can't detect it, even if it was there.
Precisely. Leads to a false negative. That's a big problem.
Okay, what about the opposite? False positives. Saying something's there when it isn't? That sounds pretty bad too, especially for rare species.
It's a serious risk, yeah. False positives can happen mainly in two ways. One is what we call allochthonous eDNA. There's basically DNA that's drifted in from somewhere else, floated downstream maybe.
So the animal isn't here, it's DNA just washed in.
Right. Or the other big source is just contamination, maybe in the lab or from the sampling gear itself if you're not incredibly careful with protocols.
And even when it works perfectly, how does it stack up against, say, actually catching and counting fish or doing detailed visual surveys? Does it tell you everything?
Not quite. It's brilliant for detecting presence or absence, like, is this species here, yes or no? But it's currently, less good at giving you the full picture.
What kind of picture?
Things like, how healthy is the population? What's the age structure, the sex ratio? Is the population stable or declining? Traditional surveys often still give you richer data on those kinds of demographic details.
Okay, right. So you've got this amazing potential, but also these significant risks and limitations, which makes this Fediajevaite study really important, doesn't it? Trying to synthesize all the comparisons out there.
Exactly. The goal was to cut through the noise and ask, does the field data actually prove eDNA is better overall for macroorganisms? And, what factors influence that?
So how did they tackle that? A meta-analysis sounds like a huge undertaking.
It was. They started with a massive literature search. Looked for papers mentioning environmental DNA or eDNA, published between 1970 and 2020. 50 years of research. And the focus just on studies looking at macroorganisms, so not bacteria or viruses.
That must have pulled up thousands of papers.
It did. But the crucial step was the manual filtering. They were really rigorous. They only kept studies that directly compared eDNA with a traditional monitoring method.
And traditional could mean lots of different things.
Oh, yeah, a whole range. Electrofishing, netting, trapping, visual surveys, analyzing scat using acoustic monitors, basically any established technique. The key was the direct comparison in the same study.
They needed that head-to-head data, not just taking someone's word for it.
Precisely. And they extracted two main types of data from these comparative studies. First, what they called quantitative continuous data.
Okay, what's that?
That focused specifically on the probability of detection. So, pEDNA for the environmental DNA method and pTrad for the traditional method.
And just to be clear for everyone listening, this probability of detection isn't just, did we find it? It's more sophisticated.
Much more. It's the calculated mathematical probability that your method will detect the species if it's actually present at the site. It accounts for the fact that detection isn't perfect, that you might miss things even if they're there.
Got it. Accounts for uncertainty. What was the second type of data?
The second type was categorical. This was simpler. It captured how the original paper authors described the comparison. Things like, was eDNA better, equal, or worse in terms of sensitivity? Was it cheaper or more expensive to detect more or fewer species?
So one set of hard numbers and one set of the author's qualitative judgment.
Exactly. That distinction turns out to be really important.
Okay, so after all that filtering and data extraction, what was the big takeaway? The headline finding?
Well, the overall conclusion, based on synthesizing everything, did seem to confirm the general consensus in the field. Where there is a direct comparison available, eDNA surveys for macroorganisms generally do come out as more accurate and more efficient than the traditional methods they were compared against.
Right, so it is better, on average, when directly compared.
Yes, and if you look just at those categorical findings, the author's own judgments, the support for eDNA looks pretty overwhelming.
How overwhelming?
Well, think about sensitivity. Out of 170 studies that compared it, 61 explicitly said eDNA performed better. For cost, out of 19 papers that assessed it, 15 said eDNA was cheaper. So qualitatively, it looks like a slam dunk.
Okay, I sense another but coming. If the qualitative story is so strong, why the need for this huge meta-analysis? Where's the catch?
Here's where it gets really interesting, as you said. The catch isn't in the details that are there, it's in the details that are missing. The field says eDNA is better, but the meta-analysis dug into the proof.
And
this is the study's big aha moment. They started with 535 relevant empirical eDNA papers that included some kind of comparison. But when they looked for truly rigorous, quantitative, comparable data, it just wasn't there in most cases.
how many actually had the hard numbers?
Get this, only 49 papers. Just 49 out of 535. That's only 9% of the relevant studies actually reported the probability of detection, the p-value, for both eDNA and the traditional method at the same site.
Wow, 9%, that's tiny.
It's a massive gap.
So this widely held belief, this consensus that eDNA is superior, is actually built on quantitative evidence from less than a tenth of the comparative work.
That's what the analysis suggests. It doesn't mean a consensus is necessarily wrong, but it means we need to be really cautious about how strong the evidence base truly is.
Why the disconnect? Why aren't people reporting the numbers?
Well, the study points out that papers were 3 times more likely to just give a general qualitative statement, our eDNA results were great, than they were to actually quantify and report the detection probabilities for both methods side by side.
It sounds like maybe it's easier to just say it worked than to do the rigorous statistical comparison.
That could certainly be part of it. It suggests the enthusiasm might be outpacing the quantitative rigor in some cases.
Doesn't that also raise flags about potential publication bias? Like maybe studies where eDNA didn't perform well or where the comparison was ambiguous just didn't get published as readily?
That's definitely a concern the authors raise implicitly. If you only publish the successes, or only the qualitative summaries of success, it paints a skewed picture. This lack of consistent quantitative reporting makes it incredibly hard to synthesize findings properly or standardize methods.
Okay, so once they isolated that crucial 9%, the studies with the hard numbers, what did that data tell them? What factors actually predict when eDNA will perform well? What drives up that probability of detection, peDNA?
Right, so focusing on those 49 studies allowed them to do some proper modeling. And a few key factors really stood out.
Such as?
Two main things, really. The specific lab technique used for amplification and the type of animal group being studied.
Okay, let's take the lab technique first. Amplification method.
Yeah, they found that using quantitative PCR, or qPCR, led to significantly higher detection probabilities compared to conventional PCR.
qPCR versus conventional PCR. Can you break that down simply? What's the difference?
Sure. Think of conventional PCR like a simple light switch. It tells you if the DNA is basically there, light on or not, light off, presence or absence.
Okay.
qPCR is more sophisticated. It's like a dimmer switch or maybe even a meter. It doesn't just tell you that the DNA is present. It tells you how much is there. It quantifies it cycle by cycle during the amplification process.
Ah, I see. And because eDNA is often present in really tiny amounts.
Exactly. Because you're often dealing with very low concentrations in water or soil samples. qPCR's ability to actually measure the quantity makes it more sensitive. You can detect weaker signals with more confidence.
Makes total sense. You need that finer tool for finding trace amounts. Okay, what about the second factor? The type of animal.
Yeah, they found a strong taxida tendency. Basically, eDNA worked significantly better, had higher sensitivity for certain groups.
Which groups came out on top?
Amphibians, insects, and invertebrates. And interestingly, these are also groups that have been quite heavily studied using eDNA, so perhaps the methods are just more refined for them.
And where did it perform less well?
It actually performed worse than traditional methods for reptiles and annelids,
Yeah.
segmented worms. And these are groups that are much less represented in the eDNA literature so far.
So maybe the methods just aren't optimized yet for them.
That seems likely. And for some groups, like mammals and mollusks, eDNA performance was statistically about the same, on par with traditional methods.
So the take-home is, don't assume eDNA will automatically be better for your specific study organism, especially if it's not one of the commonly studied groups. You might need to optimize.
Precisely. The baseline assumption should be caution until proven otherwise for less studied taxa.
Okay, what about the actual process of collecting the sample? Did things like the filter paper matter?
They did, yeah. The sampling mechanics turned out to be important. They found that the probability of detection actually increased with larger filter membrane pore sizes within the typical range used, about 0.2 to 5 micrometers. And it also increased with the total volume of water filtered.
Wait, larger pores were better? That seems counterintuitive. Wouldn't smaller pores catch more of the tiny DNA fragments?
You'd think so, but there's a trade-off. Really small pores, especially those under, say, 0.5 micrometers, tend to get clogged up very quickly with algae, sediment, just general gunk in the water.
Ah, so they clog before you can filter much water through them.
Exactly. If your filter clogs after only filtering 1 liter, you've sampled less water than someone using a slightly larger pore size who manages to filter 5 or 10 liters. Even if the larger pores are slightly less efficient at capturing the tiniest DNA fragments, filtering a much larger volume increases your overall chance of catching some eDNA, especially if it's rare.
So it's a balance between capture efficiency per milliliter and total volume sampled.
Right. You need to filter enough water to find the needle in the haystack.
Okay, so tying all this together, what are the bigger implications if the methods aren't standardized and reporting is inconsistent?
Well, the biggest challenge highlighted by these findings is that lack of standardization across the board. It's not just about filter pore sizes. The study found huge inconsistencies in how researchers even calculated the probability of detection. Some used complex statistical occupancy models. Others just used simple ratios of positive samples.
Different rulers for measuring the same thing, essentially.
Pretty much. And that makes robust meta-analysis, like this one tried to do, incredibly difficult. It also hampers replication. If someone doesn't report key details like the filter size, or whether their qPCR used probes or SYBR green chemistry, other labs can't accurately repeat or build on their work.
The whole system becomes less efficient, less reliable.
Definitely. Progress slows down.
So for researchers working with eDNA now, what's the main message from this paper? What should they be doing differently?
The authors have a very clear warning. really. They strongly caution against making just qualitative statements about performance saying eDNA was better unless you back it up with solid, comparable quantitative data.
Show your work, basically.
Exactly. The field needs to move beyond just anecdotes of success and provide the statistically sound side-by-side comparisons with whatever traditional technique is being potentially replaced. We need the numbers.
And looking forward, what specific research does the paper suggest is needed to fill these gaps and improve the rigor?
They point to a few key areas. First, we need more testing of some of the underrepresented amplification methods. Things like droplet digital PCR, ddPCR, or LAMP, loop-mediated isothermal amplification. These are promising alternatives to qPCR, but we need more comparative data on how they actually perform in different situations.
Okay, explore new tech. What else?
Second, applying eDNA more broadly.
Yeah.
We need more studies on taxa that haven't gotten much attention yet. Mammals, birds, reptiles, even plants. And also exploring different sample types beyond just water. What about sediment, snow, saliva, soil? How does eDNA perform there?
Broaden the scope, basically.
Right. And third, they emphasize the need for more explicit and comprehensive comparison criteria in future studies. Meaning, don't just report detection probability. If you're claiming eDNA is better, quantify it, provide quantitative evidence for the cost difference, the number of species detected, maybe even compare the carbon footprint of the eDNA approach versus the traditional method. Be thorough.
This has been a really fascinating deep dive. So the core takeaway for you, the listener, seems to be that yes, eDNA is a genuinely powerful tool. It's often more sensitive, often cheaper, but
the current body of published research often lacks the quantitative rigor needed to make really confident, sweeping statements that it's always better across all species, all habitats, all the time.
we've got this amazing new tool, but now the hard work is standardizing the toolbox, making sure everyone's using it, and measuring its success in a comparable way.
That's it exactly. And maybe a final thought to leave you with. This whole study focused on comparing detection presence or absence. That's where eDNA currently excels.
Right.
But a known limitation, something the paper acknowledges, is estimating actual population abundance or stability using eDNA. That's much harder. So a provocative question is, what will future meta-analyses look like when they try to compare eDNA's accuracy against traditional methods for estimating how many individuals are there, not just if they are there?
That's the next frontier, isn't it? Moving from detection to quantification of populations.
It really is. That's arguably the ultimate test for many conservation and management goals. And it's the next great challenge for the eDNA field to tackle rigorously.